Hello, my name is Max Charney, and I will be presenting my group's Enzyme lab. So we started off with our question, would a neutral pH increase enzyme activity? And we hypothesized that a neutral pH will increase enzyme activity because the enzyme, trypsin, is present in the small intestine, which has a pH of approximately 8. Therefore, we think that testing trypsin at a neutral pH will have a high energy enzyme activity. So, an enzyme is a substance in a living organism which acts as a catalyst that can create a specific biochemical reaction. Enzymes are simple catalysts made of protein. Enzymes speed up chemical reactions by lowering the activation energy of reactions. When the activation energy is lowered, it allows the reaction to happen fast. Enzymes are regulated with special inhibitors which determine which substances can bind or not bind to them. Trypsin is a protease or proteolytic enzyme which is synthesized in cells of the pancreas. It plays a role in digestion, which digests dietary proteins. It does this by catalyzing the hydrolysis of peptide bonds, breaking them down into smaller peptides and eventually into amino acids of lysine and arginine. Trypsin is active in the small intestines of humans and animals, and is secreted from gut bacteria. So to start off the experiment, we gathered our reaction buffers. We chose a range of pHs from 6 to 9, and each had a salt concentration of 0.15 moles of sodium chloride and 20 millimoles of tris HCl. The control temperature of our experiment was 37 degrees Celsius and 0.15 moles of sodium chloride because it mimics the environment of the small intestine. 500 microliters of the substrate azocasein was used for our negative control and our experimental variable. 50 microliters of our enzyme trypsin was added. We used 3 replicas of our experimental variables to achieve our results and to rule out human error, and also in case of an outlier. We only used 1 replica of our negative control variables for each pH to measure an absence of a response and to give us a baseline of our experimental values. Our negative controls were made up of 500 microliters of our reaction buffer and 500 microliters of azocasein. So after mixing the substrate, the enzyme, and the reaction buffer, we let it incubate for 20 minutes in a water bath at 37 degrees Celsius. After incubation, we added 50 microliters of 10% trichloroacetic acid, or TCA, to stop the denaturization of the enzyme and to precipitate the macromolecules, including the enzyme and the undigested substrate. We then centrifuged the test tubes at a maximum speed of 14,000 RPMs for 2 minutes to remove the undigested substrate. After the centrifuge, the undigested substrate is pelleted to the bottom of the microcentrifuge tube, and our peptide chains and free amino acids of our proteolytic enzyme suspended in this supernatant since it is not precipitated by the TCA. We then transferred the supernatant to clean cuvettes. To read the absorbance levels, we first blanked the spectrophotometer at 440 nanometers using a cuvette of deionized H2O. We read absorbances in the spectrophotometer at 440 nanometers of each negative control and of experimental value. Azocasein is a milk protein with sulfanilamide attached randomly, which allows it to absorb at 440 nanometers. So for our results, the average value at pH of 9 is higher than the other averages. We know that the difference between all of our averages are significant because our t-tests results are less than 0.05, which means that they are statistically significant. Our standard deviations are low values, which means all of our values for every trial at every pH are close together and there are no serious outliers. We did not have a t-test value for our negative control because we only performed 1 negative control at each pH. So to conclude our results, Our experimental design and execution was adequate to test the hypothesis because we used control variables that were based off of real-life conditions. Only one group in our section tested temperature, and they recorded an optimum of 47 degrees. If their data is accurate, our experiment may have reached a higher optimum if we used 47 degrees Celsius as our control, as opposed to the 37 degrees Celsius that we used. According to our conclusion, it would be the highest optimum to grow bacteria at a pH of 9, as they tend to have the highest production rates. Group 1 and Group 4 showed similar results to ours. Group 4's highest absorbance was at pH of 9, and Group 1 had a highest absorbance at pH 8. Our results do not support our hypothesis. Our values are statistically significant and showed a high value of pH of 9. Our t-test showed that these values are correct, and therefore our hypothesis was incorrect. Our sources of variability could be due to human error or laboratory mishandling. In the future, we will test more than one negative control. Thank you.